So cells have a tendency to sometimes form colonies or tissues. Colonies by just clumping up with one another, or forming tissues where, they're all kind of clumped up together serving the same function. So cell recognition can lead to the formation of a colony that's just one specific cell type, identifying another and clumping up with it. We also have cell adhesion, which is going to form, colonies or tissues. And that's where you actually get a strengthened attachment between them. So things like your desmosomes, those are going to be, it's going to be kind of like Velcro. You also have tight junctions that kind of threads your cells to one another, or you could say it sews. It's a it's an embroidery between two cells. And you also have these things called gap junctions. That's actually going to, if you notice here, they're kind of like little channels or little pores that connects the cytoplasm of cells together. So your types of cell junctions. So we're going to talk about different types of cell junctions. We have tight junctions, we have desmosomes, and we have gap junctions. So here we have tight junctions. Those are going to function roughly as kind of like a waterproofing. You're preventing, when you have a tight junction between cells, what you're doing is you're kind of preventing the movement of things between them. Your desmosomes, you're going to typically find these in areas where you need a lot of durability. Things like your bladder, you're going to have a lot of desmosomes that really form tight bonds between cells. And then lastly, you have your gap junctions. That's going to help with merging the cytoplasm so you can actually better communicate between neighboring cells. So if you need to like send signal from one cell to another, well, you might be able to send them through gap junctions. So your tight junctions, like I said earlier, that's the function of those is to prevent intracellular or intercellular movement by decreasing the space between the cells. So it's going to decrease the fluidity in the membrane slightly. And fun fact, all of our vasculature is actually composed of individual flattened cells that are embroidered to one another through tight junctions. So like I mentioned earlier, tight junctions function kind of as a waterproofing, you could say a substance proofing. The intention is to prevent substance from moving in between the cells. And so if you look closely at any of your vasculature, you'll notice that they're just a bunch of flattened cells that kind of join together to kind of make a tube in which you send plasma and blood and all kinds of other substances through your body. Now, desmosomes, like I mentioned earlier, those are going to provide very strong adhesion, kind of like Velcro. You're typically going to find this in tissues where it's crucial to increase durability. So you're going to find this in like your heart, for example, your bladder, a lot of stress. Your bladder receives a lot of stress, and same with like your gastrointestinal tissues, as well as skin. That's another one that you're going to find lots of desmosomes. So they reinforce cell connection, increase extracellular space, and they're going to be composed of, you could say, dense plaques, which attach to the cytoskeleton. So cytoskeleton, if you remember, we've got actin, we have intermediate filaments, and it's actually going to be mainly composed of intermediate filaments. So intermediate filaments are going to increase that binding strength. And then lastly, we have our gap junctions. So those are going to be, like I mentioned earlier, channels that allow communication between cells. It merges the plasma membrane between two cells. The channels themselves, we call those connexons, and those channels themselves, or those connexons themselves, are actually composed of proteins called connexins. So connexins compose the connexons. All right, so now let's talk a little bit about membrane transport. So we have passive transport, and as we know, passive transport requires no energy. So this is going to be typically just simple diffusion or facilitated diffusion, or in the case of water moving through a semi-permeable membrane, osmosis. So passive transport is just the flow of whatever substance, whether it's solute or water, down its concentration gradient. So for example, simple diffusion, that's just going to be where you have, let's just say, water, you could say a cup of water, you drop a sugar cube into it, and through simple diffusion, the sugar is going to disperse evenly throughout the water over time. Even if you had a membrane in between, let's say it allowed sugar to move through it, the sugar is still going to move from where there is a high concentration of sugar to where there is less of self or where there is less sugar. So it's going to diffuse. So let's say you have a beaker. Ooh, that's kind of weird. Full of water, and between or within this beaker, you actually have a semi-permeable membrane down the middle. And both of them have a certain concentration of ions. Ions that actually can't escape or move from one location to another. So we could say you've got a certain amount of ions there, and you got more ions over here. And you're also going to have water. Water in both chambers. Now, if you sit for a little bit of time, remember these, like I mentioned, these ions cannot move in between each of these chambers. So if you let it sit for a while, what you're going to end up with, again, this is osmosis. Osmosis only infers the movement of water. Oh, kind of just made the top for me there. Oh, I still have to make the, again, semi-permeable membrane. These are ions, so they're not going to have a tendency to move between. And then you had some concentration of ions on one side. You had a high concentration of ions on one side. And then a low concentration of ions on the other side. And so typically what you're going to have happen is the water will decrease in level on one side and increase in level on the other side. So you're going to have water that moves in this direction. Water will move in the direction where there is more solute. So water moves to where there's more solute with osmosis. Now, facilitated diffusion is going to be kind of like simple diffusion, but let's just say hypothetically, we won't even use this as an example. Facilitated diffusion is going to involve some kind of transport protein, and let's just say glucose is too big to make it through a membrane, but you have a large amount of glucose on the outside of the cell and a small amount of glucose on the inside of the cell. Well, if you put a channel up that allows glucose to flow through, down its concentration gradient from the outside of the cell where there's more glucose to the inside of the cell where there's normally less glucose, that would be a form of facilitated diffusion, because glucose would move from where there is a higher concentration of self to where there is a lower concentration of self through a facilitated transport. Now lastly, we have active transport, and active transport involves the use of some kind of energy, whether it's ATP or it could be some other type of potential energy, like a simple concentration gradient, or the movement of, you could say the movement of, an electron down your electron transport chain, which is going to drive forward the proton pumps, for example. That's another form of active transport. Not necessarily using ATP, but you're using some form of potential energy to move something against a concentration gradient. You're moving against the river, the flow or the current of the river. You're walking upstream against a river. That's going to be active transport because it's going to take some kind of energy to move against a concentration gradient. So it's kind of going against nature. So active transport is going to involve some kind of energy use. Now we also have endocytosis and exocytosis. Endocytosis is going to be the bringing in of materials, And exocytosis is going to be the excretion of materials when referring to a cell. So moving stuff into the cell or moving stuff out of the cell, it's going to be an energetic process. Okay, so we're going to talk about simple diffusion and the things that can influence the rates. So temperature is one thing that can influence the diffusion rate across the membrane. If it's warmer, typically you're going to have a higher rate of diffusion. Next is going to be the size of the molecule. Typically smaller molecules have an easier time diffusing through a membrane. And then if it's nonpolar, it's going to have an easier time moving from one side, let's say extracellular, outside of the cell, to the inside of the cell. If it's nonpolar, it's going to have a very easy time moving through the membrane. If you are sending current through the cell, a charged current through the cell, or if you have a, let's say, a variance in charge from the outside of the cell to the inside of the cell, typically you're going to have an easier diffusion rate or a faster diffusion rate in either direction, depending on where the charge is located. And then lastly, we have concentration gradients, and this one kind of makes intuitive sense. If you have, let's just say, a lot of some kind of substance on the outside of the cell, and like none of inside of the cell, it's going to have a very steep concentration gradient, meaning the difference between the outside and the inside is very large. So the larger that difference, the steeper the concentration gradient, the faster it's going to move across that membrane. All right, and so this is probably going to be some kind of review here. Tonicity, so that's just going to be the way we refer to the concentration of ions in a solution relative to what's inside of a cell. Typically, when we talk about tonicity, we're talking about the solution itself relative to the concentration of substance on the inside of the cell. So it's a way to compare two solutions. And osmotic movement. Osmotic movement is just the movement of water. So if you have a hypertonic solution, meaning you have a large concentration outside of the cell, so it looks like the peach color is outside of the cell. So if you have a lot of solute, we'll just say salt outside of the cell, water has a tendency to move from where there is a lower solute concentration to where there is a higher solute concentration. So water has this tendency to dilute stuff. And then stuff also has a tendency to want to be diluted. So if we're talking about diffusion, that stuff is going to move to where there's less of self. It wants to dilute itself. And then in the case of osmosis, where that substance can't move from one end of the membrane to the other, or one side of the membrane to another, water is going to do the job. Water is going to move from where there's less solute to where there is already more solute, with the attempt of diluting whatever substance it is. So in this case, if you have a high concentration of ions on the outside of the cell, an animal cell, for example, what's going to happen is it's going to dehydrate that animal cell. All that water is going to be dispelled from the cell. It's going to shrivel up if you're dealing with an animal cell, which don't have cell walls. Now, if you're dealing with a plant cell, what's going to happen is the plant cell will shrivel up. However, the cell wall stays intact. And so in this case, you get kind of like soggy lettuce, for example. Kind of wilted lettuce. Now, if we're dealing with an isotonic solution, iso meaning same, you have the same ion concentration on the inside as the outside, that means you're going to have a net zero change. You're going to have just as much water moving into the cell as you are going to have moving out of the cell. Same with the plant cell. So you're just going to have a normal plant, not necessarily too crisp, but not necessarily too wilted. So hypertonic refers to the outside substance having a hyper concentration, hyper meaning high or a lot. So a high concentration relative to the inside. Iso means same, so that means you have the same concentration of solute on the outside as you do on the inside. And then the very other end of the spectrum, we have hypotonic, meaning you have a low concentration of solute on the outside of the cell relative to the inside of the cell. Now, this can be dangerous for animal cells because, again, we don't have cell walls to help maintain the integrity of our plasma membrane. And so what happens is if the concentration of solute on the inside of the cell is high, water is going to move into the cell. And because we don't have that structural integrity of the cell wall? Well, sometimes if we're dealing with, our red blood cells, for example, those red blood cells might burst. That's why we don't give IVs with distilled water. Now, in plant cells, water is going to be moving into that central vacuole within the plant cell, and that's where you'll get the crispy iceberg lettuce, for example. All right, so let's talk about facilitated diffusion. So this is always going to use some kind of channel or carrier, usually composed of a protein. We have channel proteins, which are polar integral membrane proteins. We could just say integral membrane proteins. They're going to traverse the entire membrane because you're going to be moving substance from one end to another. The first one we can talk about are ion channels, and you could either have an ion channel that is just simply always open, allowing ions to flow through, or they could be gated, meaning they're waiting for some kind of cell signal or messenger to open up the channel. So before we want any ions in the cell, your channel will be closed. But then let's say it's the opportune time for ions to flow in. Let's say, for example, signal transduction. Let's say you're trying to communicate, between multiple neurons. Well, if a messenger binds to or a ligand binds to a receptor, it might cause a conformational shift in that protein, opening up the channel, allowing ions to flow in or allowing ions to flow out, for example. It can move in either direction. So ion channels is one that's just going to move ions from one side of the cell to another, and they can be open perpetually or they could be gated, meaning they require some kind of ligand to open them up. Okay, so still talking about facilitated diffusion, still talking about some kind of channel proteins. We also have aquaporins. Aqua referring to water, porin meaning you could just say it's like some kind of hole. It's like a channel. The function of this is just to allow water to move in or out of the cell. An aquaporin is just a water channel. So it's open. Likely the inside is going to be polar because like dissolves like. Polar stuff likes hanging out with polar stuff. It's going to be really easy for water to flow in or out of an aquaporin channel because the inner tube, you could say, the lumen of this channel is going to satisfy the energetic requirements of water to move through it. So it's going to be easier for water to move through an aquaporin than it is through a plasma membrane, for example. Because remember, within the plasma membrane, you still have some hydrophobic areas as well. But within an aquaporin channel, water can just easily move through. All right, so next we have carrier proteins. Those are going to bind the transported substance. In this case, we use glucose as an example. Sometimes they can be bidirectional, meaning you can move stuff into the cell, you can move stuff out of the cell. In this case, we could use glucose as an example. So it looks like we have a lot of glucose on the outside and we have very little glucose on the inside. And so glucose is just going to bind to this protein. This protein changes in conformation and then releases it into the inside of the cell. Now the thing is, eventually, if we did this and we didn't metabolize this sugar, for example, we would reach some equilibrium where the glucose on the outside is equal to the glucose on the inside. The way we maintain that concentration gradient so that there is less glucose on the inside of the cell than there is on the outside is we just metabolize the substance upon entrance into the cell. So we'll break down the glucose. We'll attach a phosphate to it. We'll undergo glycolysis, breaking it down into pyruvate molecules, for example. And then in that case, the concentration of glucose on the inside of the cell is going to be lower than what you have on the outside. And so you're still going to have that affinity for glucose to move into the cell. Now let's talk about a little bit of active transport, active requiring ATP or some kind of potential energy, any kind of energy. Active transport is going to move substance against a concentration gradient. Now, you've probably heard of uniports before. You might not have heard of the term, but the uniport, what that's going to do is it's just going to simply move one substance, uni, meaning one substance from one area against its concentration gradient into another area. So it's unidirectional, one ion type in this case. So a symport is going to move substances in a unidirectional fashion, so in one direction, but it's moving two ion types. So symport. Symport moves two different ions in one direction. They're coupled. So let's say, like this orange one and this green one. Let's just say, your orange one has like a large amount on the outside and maybe a small amount on the inside. That would be moving down its concentration gradient. But let's say this green one, there's more green on the inside than on the outside. Well, that's going to actually take energy to move this from the outside to the inside. However, the benefit of a symport is that you could actually use that potential energy of the concentration gradient of this orange one and its desire to move into the cytoplasm. You can use that potential energy to actually drive forward the movement of this green one against this concentration gradient. So if the affinity for this to move into the cell outweighs the affinity for this to move in the opposite direction, well, then you're actually going to get a nice symbiotic transport to move them both onto the inside of the cell, assuming you want more of this on the inside of the cell than you already have. Now, next one, we have an antiport, kind of functions similar to a symport, in which you could be moving two things in a direction, and maybe only one of them is moving against their concentration gradient, and the other one is moving down its concentration gradient. Or maybe both of them are moving against their concentration gradient, and you're using some other energy source to drive forward the process, like ATP. So in this case, you have an antiport, meaning they're moving in like anti-direction and in different directions. And so you have some kind of ion here moving into the cell, and it could potentially be gated or waiting for both substances to be ready to be moving in opposing directions before the transport process actually happens. Now, lastly, we have a little bit more of a complicated topic. It's called secondary active transport. And so we have a sodium-potassium pump here. So you have the active movement of one substance against its concentration gradient, sodium. So sodium, you already have a lot of sodium on the outside of the cell, but we want to put even more on the outside of the cell. So we're going to use ATP. We're using energy to drive forward the movement of sodium against its concentration gradient upstream against the current of the river, and now you have this abundance of sodium on the outside of the cell. Simultaneously, you're moving potassium into the cell. Now what you might do is you might utilize this high sodium concentration on the outside to drive forward the movement of another substance, like glucose, for example. Let's say you already have enough glucose in the cell. Let's say it's like isotonic in relation to glucose. Well, maybe your cell still wants more glucose inside of the cell. So maybe this glucose is a little bit reluctant to move into the cell. However, because you have this symport, you're actively utilizing the concentration gradient of the sodium on the outside. So the disparity between the outside and the inside is going to drive forward this sodium to want to move into the cell along with glucose. So it's going to outweigh the reluctance of glucose to move into the cell. So the passive movement of sodium along its concentration or electrochemical gradient is going to serve as an active force to aid in the glucose transport. So in this case, you're investing ATP to create a concentration gradient, and then you're utilizing that concentration gradient actively to move something against its concentration gradient. So you're creating that potential energy through creating a concentration gradient. All right, so now we're going to talk about endocytosis. So endo, you're moving stuff into the cell. So endocytosis, you have, some kind of invader or microbe. It binds to maybe receptors on the surface of your cell, or binds to some kind of protein that signals for the cell to engulf it. And in this case, you engulf it and you place it into a vesicle. Now, if it's like an invader or you want to digest it, or if it's food, you're going to fuse this with a lysosome. You're going to fuse that with a lysosome to, the lysosome containing the hydrolytic enzymes to eventually break it down. Now let's talk about exocytosis. Exocytosis is allowing things to exit the cell. So you have a secretory vesicle, which is just something to be secreted. substance to be secreted, what you're going to have are maybe signaling molecules on the surface of that secretory vesicle, which fuse with the plasma membrane and then allow it to release its contents outside of the cell. So that stuff exits the cell. Exocytosis exits the cell. So endocytosis, there's a few subcategories of endocytosis. You have phagocytosis, so that's going to be engulfing large solid particles. Pinocytosis, you could think of Pinot Noir, you're engulfing liquids. So pinocytosis is engulfing liquids, whereas phagocytosis is engulfing solids or larger particles. And then you also have receptor-mediated endocytosis, and that's kind of what I was talking about before. Let's say you have a certain ligand or signaling molecule on the surface of some kind of microbe, for example. Let's say it binds to or it interacts with a certain receptor site that would signal for your cell to eventually engulf it and then package it into a vesicle once it enters the cell. So that's going to be specific phago or pinocytosis.